Hi, my name is Cassidy Heiner. I am an undergraduate at the University of Arizona. I work in Dr. Carrie Cooper's lab. My poster is on the survival of salmonella in plant-based meat products, as well as the impact on the microbiome. The type of salmonella that was used was Salmonella typhimurium, which is one of the top 3 serovars in the world. It is mainly associated with meat. However, very little of it is known on its effects on plant-based meats. With their increased popularity, it's really important to understand if typhimurium is a potential risk to the general populace. 3 different plant-based meats were used in the experiment, as well as ground beef for control. Each type was separated into 2 groups, and they were inoculated with a 10 to the 4th CFU per gram or 10 to the 7th CFU per gram of salmonella, labeled as low and high respectively. In the 3rd group, PBS was added as a control. They were stored at 4 degrees Celsius and all sampled on days 1, 7, 10, and 14 for microbiome. They were also put into XLT4 plates to confirm or deny the presence of salmonella. The data collected is shown here in figure 1. From the XLT4 plates showed that salmonella survived throughout the 2-week period. Every single meat type and each inoculum level, except for the PBS control, had salmonella colonies on day 14 as shown. Figure 2 shows a heat map of all of the bacterial families found in the microbiomes of the meat projects without the addition of salmonella. The darker boxes indicate more bacteria of that family than the lighter boxes. There are obvious similarities between each individual meat type. However, there is overall not a very high significance difference between each plant-based meat as well as the ground beef. This lack of difference is further proved by the Faith PD and Shannon diversity plots of figure 3. No significant differences were found between the phylogenetic diversity or the diversity and evenness of the bacterial species, respectively. However, in figure 4, when looking at the phylogenetic diversity of the 3 different inoculum types, low, high, and none, there is a highly significant difference between them. The high salmonella inoculum being the lowest in diversity, and the inoculum with no salmonella being the highest in diversity. In conclusion, the Salmonella typhimurium can survive plant-based meats for at least 14 days. Also, the microbiomes of all meat types may not be significantly different from each other in the beginning, however, are highly statistically different when the Salmonella is added. Going forward, the microbiome data is going to be compared and looked at more in depth, and there will be additional experiments with other foodborne pathogens. I would like to acknowledge and thank the authors and the other members of the Cooper Lab for their support and assistance with this project. Thank you.